Hello, we are Ella
and Junyi,
and today we are explaining meiosis. Meiosis is a necessary function for sexually reproducing organisms that allows for variation and diversity in a population. It allows organisms to sexually reproduce and create offspring that are different from them. The process creates 4 haploid daughter cells, called gametes, from one diploid parent cell. Meiosis is preceded by interphase, which is the longest phase in the cell cycle. Its main purpose is to duplicate the DNA and get the cell ready for meiosis. This phase is split up into 3 parts, G1, S, and G2. G1 is essentially a growth phase, where the cell grows and develops. Once it reaches a critical cell-to-surface area ratio, it reaches a checkpoint where it can either divide, die, or stop growing. If it passes this checkpoint, it is committed to dividing and transitions to the second phase, S phase. S phase is where DNA synthesis occurs. The DNA gets copied, with each chromatid attached to its copy, also known as the sister chromatid, at the centromere. Once the DNA is fully replicated and sister chromatids have formed, the cell moves on to the final phase of interphase, G2. G2 is the last phase before meiosis, so the cell is preparing for division. Cytoplasmic organelles also replicate during this phase. After passing a final checkpoint, the cell is ready to divide. There are 2 parts of meiosis, meiosis 1 and meiosis 2, with meiosis 1 occurring first. Thus, the first phase of meiosis is prophase 1. In this phase, chromosomes condense and become visible. Chromosomes occur in pairs, 1 per parent, and are called homologous chromosomes. Homologous chromosomes stick together in pairs during a process called synapsis, 4 sister chromatids in all, creating a tetrad. Homologous chromosomes trade parts of their DNA with each other, a process called crossing over. Also occurring during this stage, the nuclear envelope breaks down. Next, the cell moves into metaphase 1. Centrosomes, which are organelles that were duplicated back in interphase and are made up of centrioles, move towards the 2 poles of the cell. Spindle fibers form from the centrosome, creating a spindle apparatus, and attach to chromosomes at the kinetochore of the centromere. The fibers guide the tetrads to the metaphase plate where they eventually line up. Each pair of homologous chromosomes get attached to a microtubule. Next, anaphase occurs when homologous pairs, but not the chromosomes themselves, separate and move towards opposite sides of the cell guided by spindle fibers. The cell elongates as microtubules push the poles of the cell further apart. By the time telophase 1 occurs, the homologous pairs are at the 2 opposite ends of the spectrum. The cell continues to elongate as the nuclear envelope reforms around the separated homologous pairs. As this is happening, the cytoplasm begins to divide in a process called cytokinesis. A cleavage furrow forms on the midline of the cell, which allows the cell membrane to divide fully. This creates 2 separate daughter cells, but meiosis is not over yet. Now, the cell moves on to meiosis 2. Unlike meiosis 1, meiosis 2 is not preceded by interphase. Instead, the cell goes straight into prophase 2. The newly formed nuclear envelope breaks down again, and the chromatin is condensed into chromosomes. After prophase 2 is metaphase 2, where a mitotic spindle forms and attaches to chromosomes at the kinetochore. Guided by the spindle fibers, the chromosomes move towards the equator of the cell on the metaphase plate. This time, chromosomes are lined up individually along the metaphase plate, with each sister chromatid attached to a microtubule. In anaphase, sister chromatids of each chromosome separate and start to move toward the 2 ends of the cell. Once they are completely separate, the sister chromatids are now considered separate chromosomes. At telophase, the nuclear envelope reforms around the 2 poles and the spindle fibers disintegrate. Finally, during cytokinesis, the cytoplasm divides and creates 2 haploid daughter cells per cell for a total of 4 daughter cells. Depending on the sex of the organism, either oogenesis or spermatogenesis occurs. Eggs are the gametes of females, where sperm are the gametes of males. In both oogenesis and spermatogenesis, the cell starts meiosis as a diploid cell. After meiosis 1 occurs, in oogenesis the cytoplasm is unequally divided between the 2 daughter cells, while in spermatogenesis it is equal. After meiosis 2, spermatogenesis yields 4 sperm with equal cytoplasms, while oogenesis creates 1 egg cell with the majority of the cytoplasm and 3 protobodies that have less cytoplasm.
Thanks for listening, and we hope you enjoyed.